Okay, so welcome to this next video in which we are discussing the adrenoreceptors. Okay, so in this video what we want to do is continue our discussion of the beta-1 adrenoreceptor and its effects on the heart. And we want to look at the way in which the beta-1 adrenoreceptor increases the force of contraction of the heart. Okay, and it does this by acting on every single cardiomyocyte in the heart, and basically what it does is it makes every single cardiomyocyte contract with a greater force, basically, and then obviously when you sum them all together, that means that the overall heart will contract with a greater force. Okay, so what I want to first do is discuss the contractile machinery within cardiomyocytes, and it's very similar to what we have seen within smooth muscle cells, so it won't be necessary to go over it in too much detail because most of it we'll just be able to draw on from our knowledge in smooth muscle cells. Okay, and then what I want to talk about is excitation-contraction coupling. Okay, so firstly, what I just want to say is that there is a difference between the structure of an atrial cardiomyocyte and the structure of a ventricular cardiomyocyte. So this is an atrial cardiomyocyte, and let me now draw you a ventricular cardiomyocyte. Okay, whoops, myocyte, there we go. Right, a ventricular cardiomyocyte, the main difference is that it has projections, finger-like projections of the membrane deep into its cytoplasm, and these projections are known as T-tubules. And atrial cardiomyocytes do not have these finger-like projections into the cytoplasm. Okay, so that's one of the key differences between atrial cardiomyocytes and ventricular cardiomyocytes. So these processes, these finger-like projections of the membrane into the cytoplasm, they are called T-tubules, and the T here stands for transverse because these finger-like projections of the membrane inwards are perpendicular to the surface of the membrane, basically. So another word for perpendicular is transverse. Okay, right, so let's now discuss the contractile machinery that you have within a cardiomyocyte. So basically you have loads of what are known as sarcomeres, okay, which are the contractile units of cardiomyocytes and also of skeletal muscle cells. Okay, so basically the structure of a sarcomere is very similar to those contractile units that we saw in smooth muscle cells. So you have two structures, okay, like so, that are equivalent to the dense bodies that we saw in smooth muscle cells, and these are what are known as Z discs. Okay, so these are the Z discs, and basically the Z discs have actin filaments which project from them towards the Z disc opposite. Okay, so here are these actin filaments, and then this Z disc over here will have actin filaments which project towards the opposite Z disc as well. Okay, so it will reciprocate like so. So these are all actin filaments. So let me just colour them all in. Okay, so we'll have these actin filaments in red here. Okay, and then suspended between the actin filaments, you then have myosin filaments. Okay, and I'll show these in a moment once I've finished colouring in my actin filaments. So here are the actin filaments, and now suspended in between the actin filaments, what you then have are myosin filaments. Okay, so you'll hopefully agree that this sarcomere structure is exactly analogous to the contractile units that we had within smooth muscle cells. Okay, so I will colour the myosin filament in blue here. And again, you can divide the myosin filaments into two halves, okay, and each of the two halves will have the myosin cross bridges oriented in the opposite direction. Okay, so the myosin cross bridges on this half here will be oriented so that the arms and then the heads point in this direction towards this Z disc, and on this side over here and they will be oriented in the opposite direction so that the cross bridges, the arms and then the heads, point towards this Z disc over here. Okay, now another thing that is different about cardiomyocytes compared to smooth muscle cells is that these contractile units are going to be very, very orderly positioned within cardiomyocytes, okay, whereas it was just a bit of a meshwork in smooth muscle cells. But in cardiomyocytes, what you're actually going to have is you're going to have sarcomere after sarcomere after sarcomere, basically. So, let me try and show these. Okay, so I might have to reduce it down to just showing 2 actin filaments from coming from each of these Z discs and 1 myosin filament, otherwise it's just going to get too crowded. So here are the actin filaments, like so, and then suspended in between them you'll have myosin filaments. Okay, so let me try and show these. So in blue, these are myosin filaments, okay, and then in red, these are all actin filaments, like so. Okay, and then you'll have means by which the actin filaments can attach to the membrane, just like we had in skeletal muscle cells. Okay, so you'll have attachment points for actin filaments on the membrane, like so, and you'll have sarcomeres like this. So basically you create these whole rows of sarcomeres within the cardiomyocyte. Okay, and it won't stop there, you'll have another one underneath this. So you'll have another row of sarcomeres underneath this. Okay, and you can just continue this drawing on, like so. Okay, so if I just draw a bit more here, so we'll have actin filaments projecting from here, 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 and all over the place, basically. And like so, and then in between those you'll then have myosin filaments in blue here. Okay, and what you can see is that the Z discs of these different rows of sarcomeres, they all sort of line up, and what this ends up creating is these striations, basically. Okay, basically, where the myosin filaments are, the myosin filaments are much bigger and much more dense. So basically, these columns, like so, where you have the myosin filaments present, okay, these are known as the dark bands, okay, and they appear dark, okay. So if I actually look at a cardiomyocyte under an electron microscope, or under just a light microscope actually, what you see is dark bands, like so, interdispersed with light bands, and basically these dark bands are called the anisotropic bands, and it turns out that these anisotropic bands are the bands corresponding to these columns of myosin filaments. And then the lighter portions, okay, so this portion here, where you don't have the myosin filaments, these are known as the isotropic bands. Okay, so these lighter portions in between, these are the isotropic bands. So you have anisotropic band, isotropic band, anisotropic band, and they alternate like so. Okay, so that is the contractile machinery then within our cardiomyocyte. What we now want to talk about is how we initiate contraction within a cardiomyocyte, because we've seen how you initiate it in a smooth muscle cell. You get the phosphorylation by myosin light chain kinase of the regulatory myosin light chain. What's very different in cardiomyocytes. In cardiomyocytes, but not in smooth muscle cells, you have troponin, okay? And this is the major difference. So, basically, in cardiomyocytes, what you have are tropomyosin molecules that lie over the actin filaments. Okay, so here is the actin filament, and now wound around it, what you're going to have is a molecule that is known as tropomyosin, like so, which will wrap around the actin filament, like so. So in pink here, this alpha helical structure is called tropomyosin. Now basically, it is attached to the actin filament by another protein. So let me draw a bigger picture here. Okay, so let's have our tropomyosin molecule like so, and then let's have the actin filament, which remember is this double helix of F-actin molecules. Okay, let's have that there. Basically, there is another molecule that attaches the tropomyosin here in red to the actin filament here, sorry, the tropomyosin in pink to the actin filament in red. Okay, and this is known as troponin, and troponin is actually a trimer of 3 portions. Okay, so this entire complex of 3 separate proteins, this is troponin. Okay, right, so the 3 separate portions of troponin then, the first portion is the portion which attaches to tropomyosin, as I've shown here, so the one in contact with our purple molecule. This is known as troponin T. Okay, then the one in the middle that isn't bound to either tropomyosin or actin, this is called troponin C. Okay, and this is the one which will bind to calcium when it goes up within the cytoplasm. And then this one here, which is bound to the actin filament, this is called troponin I. Okay, so basically, when calcium goes up in the cytoplasm of the cardiomyocyte, what will happen is the calcium ions will bind to the troponin C, and it will trigger a change in conformation of the troponin, which will then trigger a change in the conformation of the tropomyosin around the actin filament. Now, what's the significance of this? Well, usually, you do not get any contraction of the myosin filaments. So you don't get the myosin filaments climbing up the actin filaments and pulling the actin filaments inwards. Okay? And the reason you don't is because all of the active sites, i.e. all of the binding sites where the myosin cross bridges could bind to the actin filament, those are covered by the tropomyosin molecule. However, when calcium binds to troponin C, it will trigger a change in conformation of the troponin molecule, which will change the position of the tropomyosin over the actin filament, and that will rearrange the tropomyosin so that now active sites on the actin filament are visible, and the cross bridges of the myosin molecules can actually now bind to these active sites. So the myosin heads can now bind to the active sites, and the cross-bridge cycling process, the process of the myosin heads climbing up the actin filament and pulling the actin filaments inwards, this can now begin, and hence contraction can begin. So that's how a rise in intracellular calcium can then lead to the contraction of cardiomyocytes. Okay, so what we now want to talk about is excitation-contraction coupling. Now, we will do this in a ventricular cardiomyocyte. In atrial cardiomyocytes, it's the same, but everything occurs on the membrane, on the peripheral membrane rather than on the T-tubules. Okay, but so we'll draw this for the T-tubule here. So this is a ventricular cardiomyocyte. So excitation-contraction coupling, which is often just abbreviated to EC coupling. Okay, and I'll just write out EC is just short for excitation-contraction coupling. This is the process by which you couple the electrical excitation of the membrane of the cardiomyocyte to the contraction of the cardiomyocyte. So you want the fact that the cardiomyocyte is undergoing an action potential to result in the contraction of the cardiomyocyte. Okay, how is this going to happen? Well, basically, the excitation of the membrane of the cardiomyocyte needs to cause an increase in the level of calcium in the cytoplasm of the cell, and that calcium can then act on the troponin C and begin the process of contraction of the sarcomeres. Now you might think, well, that's easy, we'll just have voltage-gated calcium channels on our plasma membrane. But basically, we are going to have voltage-gated calcium channels on our plasma membrane, but the amount of calcium they let in is not enough. So we need a better system, basically. So, this is what's going to happen, basically. You will have voltage-gated calcium channels on your membrane, and they'll be on the membrane, for instance, of the T-tubule here. Okay, so I'll draw one of these, and this is a challenge, drawing it sideways. I'm beginning to regret using the ventricular myocyte as an example. If we'd used the atrial myocyte, I would have just been able to draw it up here. Okay, but never mind. So I'm going to draw a voltage-gated calcium channel. Now we've seen the structure of voltage-gated calcium channels before. They consist of this alpha-1 subunit, which is the major subunit here. Okay, and then these accessory subunits, the gamma subunit, the beta subunit, and the alpha-2 delta subunit, which is over here. Okay, so this is an alpha-2 delta subunit. Right, now the forms of voltage-gated calcium channels that you're going to have in cardiomyocytes are going to be L-type voltage-gated calcium channels. Okay, so we're talking about a normal ventricular cardiomyocyte now, a contractile one. Okay, and you have present in the membranes of these contractile ventricular cardiomyocytes and also atrial cardiomyocytes, you have these L-type voltage-gated calcium channels here. Now let me just remind you of what that means. That means that the alpha-1 subunit is within that family of CAV1, okay, and there were 4 members within that family. There was CAV 1.1, CAV 1.2, CAV 1.3, and then all the way up to CAV 1.4. Okay, so if you use one of those alpha-1 subunits, then you are considered an L-type voltage-gated calcium channel. So these voltage-gated calcium channels are going to be L-type. Okay, so when the cardiac action potential makes its way along the T-tubule, okay, and I should just say, it'll propagate along the membrane of the cardiomyocyte, down the T-tubule, and then it will get to this L-type voltage-gated calcium channel, and it will trigger that L-type voltage-gated calcium channel to open. And we know that as soon as you open a calcium channel in the membrane of a cell, it lets calcium in because of the 15,000-fold concentration gradient across the cell membrane. Okay, so you get a rise in calcium around the intracellular aspect of this L-type voltage-gated calcium channel, and that calcium signal is called, well, it's so important that it has a special name. It's called a calcium sparklet. So a calcium sparklet is the name for the rise in calcium around the L-type voltage-gated calcium channel. Now you will have these things occurring all over the place, but they are not large enough to actually cause contraction of the cardiomyocyte. So you now need to amplify them, and this is done through a structure known as a calcium synapse. Okay, so basically what you will have is a portion of the cardiomyocyte's equivalent of an endoplasmic reticulum, which is a sarcoplasmic reticulum. So basically, the sarcoplasmic reticulum is just what would be called an endoplasmic reticulum in a normal cell, but because we're dealing with a cardiomyocyte, it's called a sarcoplasmic reticulum. And this is a fantastic store of calcium, basically. Okay, and basically what you will have is a little process of the sarcoplasmic reticulum that will face onto the portion of the membrane where you have these L-type voltage-gated calcium channels. And basically this forms a structure that is starting to be referred to as a calcium synapse. Okay, because basically what happens is this is like the presynaptic membrane. It's releasing calcium into what's the equivalent of the synaptic cleft, okay? And people actually do call this gap in between these two things the dyadic cleft, okay? So this is sometimes referred to as the dyadic cleft. Then the calcium will diffuse across to the postsynaptic membrane over here and act on a receptor on that postsynaptic membrane and trigger a response. So it really is like a calcium synapse. Now, basically the receptor on the postsynaptic membrane here is going to be what's called the type 2 ryanodine receptor. And I just want to discuss the type 2 ryanodine receptor in a bit more detail. So this is the type 2, and then I'll just put ryanodine receptor, RYR, for now. Okay, so let's go over the page. So, basically, ryanodine receptors are massive great proteins. These are some of the biggest proteins you will ever encounter in biology. They are enormous, giants of protein, of the protein world. They are visible on electron microscopes. They are so, micrographs rather, they are so big. Okay, so ryanodine receptors are tetramers, so they're made up of 4 separate proteins all stuck together. Okay, so this is a ryanodine receptor. Okay, and basically there are 3 separate ryanodine receptor subunits, but before we discuss those, let me just show you the structure of a ryanodine receptor subunit, the membrane spanning topology, the usual convention. Okay, so let's pull out one of these quarters of a ryanodine receptor here, and let's have a look at its membrane spanning topology. Okay, so the membrane spanning topology of this quarter of a ryanodine receptor is as such. Okay, so you start with the amino terminal in the cytoplasmic side, and remember this is the SR membrane, so this side is the cytoplasm, and this side now is the SR lumen. Okay, right, so on the cytoplasmic side we have the amino terminal, then we have 2 membranes spanning alpha helices, a 3rd, a 4th, a 5th, a little P loop, and then the 6th. So this is the membrane spanning topology of the subunit of a ryanodine receptor, and they are massive proteins. They are around 5,000 amino acids long. That is big. That is really, really big. Okay, now there are 3 separate genes for ryanodine receptor subunits. There is the ryanodine receptor 1 subunit, okay? There is the type 2 ryanodine receptor subunit called RYR2, RY for ryanodine and then R for receptor. And then there is the type 3 ryanodine receptor subunit, okay? Now, these, as far as we know, only form homotetramers, okay? So they only aggregate together with subunits that are of the same type, okay? So for instance, you can make a ryanodine receptor type 1, which will be made up of 4 type 1 ryanodine receptor subunits. You can make a ryanodine receptor type 2, which will be made up of 4 type 2 ryanodine receptor subunits. You can make a ryanodine receptor 3, which will be made up of 4 type 3 ryanodine receptor subunits. Okay, right. We'll continue this discussion in the next video.